So, hello everyone, good evening. Let's start today's class. A short introduction about myself. My name is Kajal Gachawa. I'm a PMRF Research Scholar from IIT BHU Varanasi. I'm your TA for this particular course, that is Host-Pathogen Interaction, the duration for which is January to April 2025. And we will be solving the weekly assignments on every Saturday from 3 to 5 p.m. And today we will be solving the week 5 assignment, and we will also learn about some topics from the week 5. And the lecture material for week 1 to 6 is already released on the NPTEL portal. And if you have not visited that yet, so you can go and visit the NPTEL. And also the video material up to the week 5 is also available on my YouTube channel, so you can check it out there. This is your syllabus, and till yesterday, the week 7 content is released on the NPTEL, and also the assignment for week 7 is also there, so you can check it out. So, today we will take some topic from the week 5 material, that is about the innate immunity. We have already discussed about innate immunity in our very initial classes. This is our body's first line of defense against infections. Unlike adaptive immunity, which takes time to develop, innate immunity is always present and acts immediately when a pathogen enters the body because it is present with us from the birth. So, there are some components of the innate immunity. The first one is physical, mechanical, or anatomical barriers. That is, these are the physical barriers that prevent microbes from entering the body. The example of these type of physical barriers are like our skin and mucous membrane, cilia. Somebody is there. Hello, hi Shweta, you are there?
Hi, ma'am. I'm here, but I cannot talk.
No problem.
Can I text in
Yes, yes, sure, sure.
the
You
chat
can.
box? If
Yes, yes,
I'll
sure,
listen to
sure.
it, ma'am. Sorry, I'm very sorry. I'll
Yes,
just
yes,
listen
no
to the lecture.
problem. No problem. It's fine. You can write in the chat box. I'll get your queries from there, or you can
Yes.
listen to my lecture. Whenever you
Yes.
have any doubt, just raise your hand or drop a message in the chat box so that I can get notified about that and I will check it
Yes,
out from
ma'am.
this video. Otherwise, you
Yes,
can
ma'am.
listen to the lecture. Okay.
Yes, ma'am. Thank you so much.
Okay. So we have just started about the innate immunity, which we have already discussed in our very initial lectures. Like this is the first line of defense against infections. So unlike adaptive immunity, this innate immunity is always present with us and it acts immediately when a pathogen enters the body. And this is present with us from the birth. So we were discussing about the components of innate immunity. So first one is the physical, mechanical or anatomical barriers, which consist of skin, mucous membrane and cilia. And they act like a wall or the shield, like they act like a wall or the shield for blocking the harmful microbes from getting inside. Like they are the first line of defense of our body from the pathogens. And the second barrier is biochemical or chemical barriers. These are the natural molecules in our body that kill or slow down the microbes. So there are some examples to this. The first one is like MBP, that is mannose binding protein. It helps to recognize and destroy the microbes. The second one is CRP, which is C-reactive protein. It signals for immune cells to respond for the infection. Third one is APP, that is acute phase proteins. These are the proteins that increase in our body when we encounter an infection. And the next one is serum amyloid A and alpha 1 antitrypsin and celluloplasmin. These are the molecules that help control the inflammation and the infection in our body. The next molecules are the human beta defensin and lysozymes. So these are responsible for direct killing of bacteria by breaking down their cell wall. So these are responsible for that. So what we discussed just that we have some biochemical or chemical barriers to protect us from the infection. The first one in that is the antimicrobial peptides or protein, example of which is MBP, mannose binding protein, C-reactive protein, acute phase protein, and some of the serum amyloid A, alpha 1, antitrypsin, celluloplasmins, and some human beta defensins and lysozymes. So these are responsible for killing the pathogens and not allowing them to enter in our body. So the like the first we have discussed the physical, mechanical or anatomical barriers. They act like a wall to protect us from the infections or from the harmful pathogens. The next one is biochemical chemical barriers. These are like chemical weapons. So these are like chemical weapons to protect us from the pathogens or the infection. And this next one is complement. What are complement? Like these complements are a group of proteins that work together to destroy bacteria by making holes in their membrane. Like they form pores in the membrane of the pathogens and destroy these pathogens. So they help the immune cells in recognizing and attacking the pathogens faster. So this is the function of complements. The next one is microRNAs against viruses. And what are these? These are the small genetic molecules that help block viruses from replicating inside our cells. So these are responsible for blocking the viral replication in our body. So they act like a molecular scissors for cutting down the virus ability to grow and the spread. So these microRNAs are the molecular scissors which prevent the viral replication and their spread in our body. So the next one is the third one is microbial or microbiological barriers. What are these? Like you might have heard about the good bacteria of our body. So these are that only. The microbial or microbiological barriers are the friendly bacteria or the good microbes that live in our body, in our gut, skin, mouth. Like we can find some gut bacteria, skin microbiota. So these are the good bacteria which live in our body and prevent harmful microbes from growing, because these good bacteria can compete for the nutrients and the resources available for their growth, so that the harmful bacteria cannot grow and cannot spread infection in our body. So what are these? These are the good bacteria which prevent the growth of the harmful bacteria. They don't allow the growth of this harmful bacteria. So these good bacteria are the microbiota. Like we have heard about the gut microbiota, skin microbiota. So these are responsible for preventing the infection from pathogens. The fourth one is the cellular barriers. The cellular barriers, as from the name you can see, these are the immune and non-immune cells of the body. Like these are the cells which help in fighting from the infection. So example of this is NK cells, that is natural killer cells. It kill virus infected cells and some of the other pathogenic cells of the body. The next one is macrophages. We have already discussed so many things about the macrophages, like these are responsible for the phagocytosis of bacteria and the dead cells in our body. So macrophages also protect our body from the infection. Next one is the dendritic cells. These dendritic cells act as the messengers and they inform other immune cells about the infection, so that collectively all the immune cells can fight from the infection. So there are some cellular barriers for protecting us from the infection, which are part of our innate immunity. So this is from the innate immunity, the components of innate immunity. First one is the physical or mechanical barriers, which act as a wall to protect our body from the infection. Example of which is skin, mucous membrane, and the cilia. The second one is biochemical or chemical barriers. Like these are like the chemical weapons to protect from the infection. The type of which is antimicrobial peptides or proteins, the MBP, CRP, APP, and many others. And the other type of that is complement protein. The next type is microRNA. These microRNA act as molecular scissors for because they kill the because they stop the virus replication. So these are the molecular scissors. And the next third one is the microbial or microbiological barrier. That is the gut microbiota or the skin microbiota, which are the good bacteria of our body, and they kill the harmful bacteria from the body. They don't allow their growth and spreading of the infection. The next one, fourth one, is the cellular components. These are the all the cells of the body, that is immune and non-immune cells, for example NK cells, macrophages, and dendritic cells. So these were the components of the innate immunity. So next we have this mechanical, chemical, and microbial barriers. This first one was the components of innate immunity. Now we will discuss in detail about the mechanical, chemical, and the microbial barriers of the innate immunity. So let's discuss that how our body prevents infection using different kind of barriers. These barriers are divided into these three categories. First one is mechanical, second one is chemical, third one is microbiological. So, this mechanical barrier are the physical structures and movements that stop microbes from entering. Like these will stop the entry of the microbes from the outside. And these chemical barriers are the substances that destroy or inhibit the microbes. Like even if any microbe is able to bypass the mechanical barrier and get entry in our body, then these chemical barriers will destroy or inhibit their growth. And the third one is the microbiological barrier. So these are just to discuss that the good bacteria of our body that don't allow the growth of harmful ones and spread of infection. So even if these chemical barriers are even in the case when these chemical barriers are unable to kill the pathogens, then this microbiological barrier will come into role, and it will protect the growth of harmful bacteria in our body. So like this, three type of barriers play simultaneously. So let's discuss the detail of this mechanical barrier. So mechanical barriers are like the shield or wall that we discussed there. This is the wall for protection. That is the first line of defense. So this mechanical barrier act as a shield or wall that prevent microbes from entering the body. And this first example is skin. So this what the skin do. This is very common that we are aware of this, that skin forms a tight barrier to block bacteria and virus entry. And this next one is gut. What gut do? This moves fluid and food in one direction to flush out the harmful microbes. If any pathogen is trying to enter through the gut, then it will move the fluid and food in one direction so that the pathogens are flushed outside and they don't enter in our body. The next one is the lungs. These lungs, what the lungs do, they have cilia. The cilia are the tiny hair-like projection. Cilia are the hair-like structure. So what they do, they move mucus and trapped microbes out of the airways. Like in our lung lining, there are cilia inside this. These are very tiny hair-like structure. So if a pathogen is trying to enter in our lung area, then the cilia will push out the pathogen. It will not allow the entry inside the lungs. So in this way, cilia protect our lungs from the infection. The next one is eyes, nose, and oral cavity. So these contain the tears or the nasal cilia, which wash away the microbes. Like from our eyes, we get tears, from our nose and oral cavity, we get the mucus secretion. So these also flush out the pathogen's entry. So these are the mechanical type of barrier. The next one is this chemical barrier. First one is mechanical, second one is chemical barrier. So these chemical barriers use enzymes, acids, and antimicrobial peptides to destroy pathogens. So in the case of chemical barrier, what skin will do? Skin contain fatty acids that make it hard for bacteria to survive on the skin layer. So this will not allow the entry and growth of this pathogens on the skin. So that skin have fatty acids onto it. And in the case of gut, it have a low pH and the enzyme called pepsin, which creates a acidic environment, and this will digest any harmful microbes if they have entered in the body. And the next one is lungs. Lungs create this pulmonary surfactant which helps to protect from the respiratory, which helps the inhibiting the entry of pathogen in the respiratory tract. The next eyes, nose, and oral cavity. These have enzymes in tears, saliva, which is a very common. We know that it contains the lysozymes. This is the enzyme present in the tears and the saliva. It breaks down the bacterial walls and it will not allow the growth of the pathogens. So in this way, it protects our body. So, like there are some other barriers like this microbiological barriers, which is like we have seen this mechanical barrier are the wall, this chemical. Somebody's there. But until here, everything is clear to you.
Yes, ma'am, it's clear.
Okay. Okay, so what we were discussing, the third type of barrier is microbiological barrier. That is the normal healthy microbiota of our body, that is the skin microbiota and gut microbiota, which compete with the harmful microbes and they prevent the pathogens from growing and spreading the infection in our body. So these are the three type of barriers of our innate immunity, which helps to protect us from the infection. So there are more details to the skin, that is the physical component of the defense. It have skin is divided into layers, that it have this epidermis, which is the outer layer which act as a shield against microbes. Then it have the dermis, that is the inner layer. It provides strength and flexibility to prevent the damage from the pathogens. So there are two layers of the skin. First one is epidermis, that is outer layer. Other one is dermis, that is the inner layer. So this dermis act as a shield and epidermis act as a shield and dermis provides the strength and flexibility. So this epidermis is composed of multiple layers of tightly packed cells, and it makes it hard for the bacteria and other pathogens to enter because this is very hard layer. So very few pathogens can penetrate these layers. And it also have a property of shedding off time to time, like it will shed off the dead skin cells, which also remove the attached pathogens onto it, so their entry will be prevented. And this also have epidermal dendritic cells which can phagocytose pathogens. So this dendritic cells act like this guards in protecting the infection, and it can recognize and engulf the invading microbes which can cause the infection. So these are the extended out among other cells of the epidermis, forming a network to intercept invader. Like they will phagocytose the pathogen if they are trying to enter. And the next inner layer is the dermis. This dermis layer is rich in collagen. Collagen is a protein that gives the skin strength and the flexibility. Like strength and the pliability, that is the flexibility. So that this will help the skin to resist any cut or injury onto that, so that the pathogens cannot enter. The next one is skin as a biochemical component of defense. Like this was the physical component of the defense, like how physically this can protect mechanical barrier. And the second one is biochemical component of the defense, that is the chemical barrier category. So it can, like skin have the capacity for the perspiration, that is the sweat production. So there are sweat glands which produce perspiration, which contain substances that fight microbes. Like this have salt which will inhibit the growth of pathogens by drawing water from their cells and leading to dehydration and death of the pathogens. And that's why we have sweaty skin is less prone to the bacterial infection. And the next one is antimicrobial peptides like defensin and cathelicidins. These are the small proteins that kill bacteria, fungi, viruses by disrupting their cell membrane. So this will also kill the pathogens. This defensin and cathelicidins act like natural antibiotics to prevent the infection. The next one is lysozyme, which destroys the cell wall of bacteria. And this is found, as we have just discussed, this lysozyme is found in the tear, saliva, and the sweat, which help in protection of infection. And the next capacity of the skin is sebum production. First was perspiration, that is sweating. Second one is sebum, that is the oil production. So, sebaceous glands which produce sebum, it help to protect us from the infection in two ways. Like it help in keeping skin pliable, like that is flexible, and less likely to break or tear. and it also lowers the pH of the skin to a level that can inhibit too many bacteria. So these are the chemical or biochemical properties of the skin. The next one is there is a structure of the skin, that is the physical and biochemical components of the skin. So here we can see that the skin is covering whole our body. So this is the longest first line of defense which can protect us from the infection. So there are some layers or some parts of different type of cells or different layers in the body. So we'll understand one by one. The first one is this stratum corneum. This is the outermost layer which is made up of dead skin cells and keratin. So the stratum corneum is made up of dead skin cell and keratin. So it acts as a tough barrier that prevent the microbial entry. This is the hard outside layer. The second one here, from here to here, it is the stratum lucidum, which is a thin, clear layer found only in thick skin. Thick skin is like our palm and our toe. So it is found at the that thick part of our body. So this is the stratum lucidum, second layer. The third layer is the stratum granulosum, and this contains the keratohyalin granules. Like there are some granules you can see. So it contains the keratohyalin granules, which help to form a watertight lipid layer, which helps to form a watertight lipid layer, which prevent the moisture loss and also help in prevention of microbial invasion. And the next fourth downside this layer here is stratum spinosum. It provides structural strength and it also contains some of the immune cells in it. The lower last layer is the stratum basilae, which is the deepest layer where new skin cells are formed. Here, the new skin cells are formed. So, these are the layers of the skin. The first one is stratum corneum, which is the first outermost layer. It contains dead skin cells and keratin. Second one is stratum lucidum. It is the second layer, which is only found at the thick skin part, like palm and our toes. And the third one is stratum granulosum, which contain keratinohyalin granules, which help to form the watertight lipid layer, which create a moisture environment and also protect us from the microbial invasion. The fourth one is stratum spinosum, which provides structural strength and it also contains some of the immune cells. The last one is stratum basilae, which is the deepest layer of the skin where the new skin cells are formed. So these were the physical components of the skin. And when we talk about the biochemical defense of the skin, here in case this watershed lipid layer, what is the role of this layer? This layer will prevent this yellow layer that you can see here. This will prevent the moisture loss and it will keep our skin hydrated and resistance to microbial invasion. So, and here there are some lamellar bodies. These are the small granule-like structure here. These release the antimicrobial peptides, like that we have discussed back in the last slide, also beta defensin and cathelicidins. So these are released from this lamellar bodies. What this beta defensin do? It will disrupt the bacterial membrane and kill the microbes. And this cathelicidin also have a similar function. It will help in fighting from the bacterial, fungal, and virus infection. Bacterial, fungal, and virus infection. It will prevent their replication and their division. So these are the components of the skin. So I hope until here this is clear to you. So we can move forward to this mucosal surface.
Yes, now it's clear.
Okay. So we'll move forward to the this mucosal surface. What are these mucosal surface? These line our body cavity openings to the outside environment. Like what they do, these are the mucosal surfaces found in the like this respiratory tract, digestive tract, and urogenital tract, which are the areas that are exposed to the outside environment. So these are found at the cavity openings towards the outside. That is the, I'll write it here, respiratory, digestive, and urogenital openings of our body. So these are found at those places. So this unlike the skin which have multiple protective layers, we have seen in the case of skin here, there are so many layers. So in mucosal layer, there are not so many layers. So these are itself very thick and they make the pathogen entry impossible. Like skin is very thin, so there are so many layers of the skin. Like if pathogen can enter into one layer, the next layer will prevent the further entry and so on. But in the case of mucosal, this mucosal surfaces, these are the only one cell thick. These are the only one layer that is thick, and it make the pathogen entry very difficult. So what they do actually, these are like linked to the associated with the lymphoid tissues. Like we have discussed in some previous lectures about the M cells in the gut. So these mucosal epithelial cells are linked closely associated with and linked to the lymphoid tissues. Example M cells in the gut that help in detection and transportation of antigens to the immune cells for a quick response. Like if a pathogen enters in the body, these M cells will interact with them and they will transport them to the other immune cells for the further response. And these epithelial cells are packed tightly to prevent the entry of pathogens, but often only one cell thick layer. So pathogens sometimes breach this barrier because this is a single layer. This is a single. And the other property of this mucosal surface is the continuous shedding of cells, carriers, attached microbes away. Like these will shed off regularly. Like the old cells will constantly shed off and they will also remove the bacteria or microbes which are attached to them. So they will prevent the growth of these pathogens. And besides producing mucus, mucous membrane also produce lysozymes and other antimicrobial peptides. Like this mucus will trap pathogens and prevent them from reaching the deeper tissues. And after trapping, they will produce the lysozyme and other antimicrobial peptides, which are also secreted to break down bacterial walls and kill the pathogens. So there is a fact that every day an adult human swallow and digest about 1 liter of mucus. Like from all our different parts of the body, we swallow and digest about 1 liter of mucus per day. So this mucus carries trapped microbes to the stomach where they are destroyed by the stomach acid because we have seen in the previous slides that stomach have acidic environment which will digest the pathogens if they are entered. So the mucus surfaces are also present in our lungs. So, as you know, that lungs are constantly exposed to airborne pathogens because we breathe in so many pathogens with the air. So, these lungs have developed a specialized defense system to trap and remove microbes before they can cause the harm. So, there are different type of these cells and layers present in the lungs. The inner lining of this lung is covered by the ciliated epithelial cells and the goblet cells. These goblet cells, what they do, they secrete the mucus. The function of this goblet cell is to secrete the mucus, which trap the dust particles, microbes, and other harmful pathogens along with the air which we are breathing in. And the cilia, we have seen that this cilia is the hair-like structure which constantly move in a way that it push the mucus and the trapped microbes out of the airways. Like it will sweep away if any pathogen is entering in our body. So this is the function of the cilia. It is constantly moving. These are the small hair-like projections which will move away the pathogens. So the next function of this mucosal surfaces in the lungs is this mucus is very sticky substance that traps the microbes, dust and the pollutants and prevent them from reaching into the deeper lung tissues. And this mucus also contain antimicrobial peptides that we have just seen. What are those antimicrobial peptides? Lysozyme and defensin molecules. These are the antimicrobial peptides present in the mucus, which will kill the pathogens. So, like if mucus and the cilia don't work properly, then what will happen? When these harmful bacteria and viruses can enter in our lungs, and they can lead to the infections like pneumonia, bronchitis, and other infections. So, in the cases of like cystic fibrosis, you might have heard about these terms cystic fibrosis and chronic smoking. In the case of chronic smoking, what happens that in these conditions the cilia is damaged. Cilia is damaged so that mucus cannot clear this pathogen. Cilia is damaged, so mucus will not be pushed outside. So it will increase the risk of infection. It will cause the infection. So in the case of cystic fibrosis or some like people who smoke very chronic from the chronic time, their cilia is damaged. And because of the damaged cilia, it is unable to move continuously and it cannot like push away the pathogens. So it will cause infection. So this is an example case where the cilia is not functioning properly. The next one is here this mucosal surfaces in the gut. We have seen this gut microbiota. So, what happens here? The gut lining or this gut epithelium. Here we can say this gut epithelium or gut lining. It is made up of different type of cells that work together to defend against these harmful pathogens. So there are also goblet cells in the gut which will secrete the mucus. We know the function of this goblet cell is the mucus production. So it will form a layer to protect bacteria in the same way that we have just seen. And it also have this Paneth cells. What these cells do, these are found in this intestinal cryptic region. Intestinal crypt, here these are found. So what the Paneth cells will do, they will produce the antimicrobial peptides It will produce the antimicrobial peptide, that is the alpha defensin and this reg3. So this alpha defensin will kill the bacteria by rupturing its membrane, and this reg3 will target the gram-positive bacteria and prevent them from colonizing the gut lining. So these both will kill the pathogens. And there are also some macrophage cells. These are present in this. We have studied about some tissues, that is the galled and walled. We have studied it previously. So these macrophages are present in this gut associated lymphoid tissue. It will do the phagocytosis, that is the engulf and destroy the pathogens. So in this way, the gut also contain the mucosal surface that protect from the pathogen. The next defense part is lysozyme. So this is an enzyme found in tear, saliva, mucus, and several other body fluids. So it works by breaking down the bacterial cell wall and making like these the gram positive bacteria and also gram negative. This function in both ways. Like in gram negative bacteria, the LPS layer is present, that is the lipopolysaccharide layer. So what these lysozymes will do, these lysozymes will block this. Like there is this LPS layer here, lipopolysaccharide layer present in the gram negative. It will block lysozyme from reaching the peptidoglycan layer. This will make the lysozyme action more resistance. And if this membrane is damaged from here, then this lysozyme will enter inside the body. So it can degrade the peptidoglycan layer of this pathogen. And also in the case of gram-positive bacteria, which have a thick peptidoglycan layer, this lysozyme can easily break down this peptidoglycan layer and enter inside. So it will also kill this bacteria. So this lysozyme is responsible for killing both gram-positive and gram-negative bacteria. The next defense molecule is the defensin, cathelicidin, and reg3 that we have seen. So till here, is this clear?
Yes, ma'am.
Okay. So these are the antimicrobial peptides like beta defensin, alpha defensin, beta defensin, cathelicidin, and reg3, which are responsible for killing the pathogens. These have different role. Like these defensins are the positively charged peptides that attack the membrane of these pathogens. Like there are three main type of amino acid sequences. Like first one is this alpha defensin, beta defensin, and this delta. What is this theta defensin? So these are the small molecules which are responsible for killing the pathogens. So, like we know that bacterial membranes are negatively charged, these are negatively charged, and then defensin molecules are positively charged. So, by this, they will try to enter inside this negatively charged membrane and they will create pores or the holes in the membrane. So, if bacterial membrane is created pore, then the pathogen cannot survive. So, it will kill them. So there are some more antimicrobial peptides like this lactoferrin, azurocidine, and vitamin B12 and BPI, that is bacterial permeability inducing protein. So there are many other antimicrobial peptides which are responsible for killing the viral, fungal, and parasitic pathogens. So we'll take this quickly. That is lactoferrin, which sequesters iron, that is, it binds to the iron and make it unavailable for the pathogens, for bacteria. So, in the absence of this iron, the pathogens cannot grow. So, ultimately, they will die. And this have a bacteriostatic property, that is, it stop the bacterial growth because the iron is not available for the bacteria, and in the absence of iron, it cannot grow. So, it will stop the growth of these pathogens. And it also have this bactericidal activity, that is the bacteria static is the stop the growth of this pathogen. Bactericidal is the kill the pathogens. So this also have bactericidal activity because this lactoferrin binds to the lipid A layer of this LPS, that is the lipopolysaccharide layer of the bacteria, and then it can destabilize the membrane so that pathogen will be killed. And like some bacteria also produce the siderophores. The siderophores are the molecules that steal iron from the lactoferrin to survive. So, like this bacteria have siderophore molecules. So, this iron will bind to the siderophore instead of lactoferrin. So, in that case, these can survive. So the next one is azodocerin. So it works like both the antimicrobial peptide and a chemotactic protein because it helps in attracting the immune cells, that is the neutrophils, to the infection site. So these are the azodocerin. It have the antimicrobial property, like it will kill the pathogen, and it also have the chemotactic property. It also act as a chemotactic protein. So it attract the immune cells to the site of infection. And it was originally evolved from the serine protease, but now these are the inactive enzymes for the immunity, the serine proteases. So the next one is this BPI, that is bacterial permeability inducing proteins. These are also responsible for targeting the LPS layer of gram negative, LPS layer in gram negative bacteria, and it increase the membrane permeability and cause the bacterial death. So it also acts like a natural antibiotic produced by the immune cells. So this BPI act as a natural antibiotic produced by the immune cells. So the last one is vitamin B12 binding protein. So bacteria need vitamin B12 for its growth, but this protein, this vitamin B12 binding protein, it will bind to this vitamin B12 and it will make this vitamin B12 unavailable for the bacterial growth. Same like this lactoferrin was binding with iron and making it unavailable for the pathogen's growth. In the same way, vitamin B12 binding protein will bind to the vitamin B12, and this pathogen will starve for this nutrient and they will die. Next are this biochemical barrier, which are the reactive nitrogen species and reactive oxygen species. So these are highly reactive molecules, and these can destroy these pathogens. Like there are some type of this reactive oxygen species. First one is the superoxide anion. The next one is hydroxyl radical, hydrogen peroxide, hypochlorite anion. So these are all responsible for killing the pathogens. And also the nitrogen species are nitric oxide, nitrogen dioxide, peroxynitride. These are responsible for killing the pathogens. The last one is this microbiological barrier that we have seen the gut microbiota and the skin microbiota. So these are the good bacteria of our body. These are our good friends which help us from the which help to protect us from the bad pathogens. So like there are so many pathogens, like so many good bacteria in our body. This is where you can see this in mouth, a major part is as firmicutes, and in the skin, the major part is of this actinobacteria. And in vagina, also the major part is firmicutes. In colon, also firmicutes, and equally the bacteroidetes. And stomach also major part is firmicutes, and esophagus also there are this different type of bacteria, firmicutes, bacteroids, actinobacteria, proteobacteria, and some other phyla of the bacteria. These are present in different parts of our body. So these firmicutes, these include this very common the lactobacillus and the clostridium species. This lactobacillus and the clostridium species are in the firmicutes. So this is present at major part of our body. The next one is bacteroidetes, which consist of this bacteroides fragilis. So this is mostly found in this colon area, and it is important for breaking down the complex carbohydrates and train the immune cells in fighting from the infection. The next one is actinobacteria, which is the bifidobacterium species. It is majorly found in the skin. Major part is in the skin. And the example of this actinobacteria is this bifidobacteria, and it supports the gut health and prevent infection. This next one is this proteobacteria, example of which is E. coli, which is very commonly found in most of our body, but majorly this is present in our gut. So these are some of the common good bacteria which are destroying the harmful bacteria and preventing us from the infection. So next there are a different topic that is pattern recognition receptors, that is PRR. So these are the receptors in our immune cells that detect the pathogen and trigger immune response. From the name we can see recognition. So these recognize the pathogens, that yes, there are some pathogen entry in our body, and they will call upon the immune cells. They will tell the immune cells to take action and protect us from this harmful pathogen entry. So these PRR are the unique molecular signatures which are found only on the microbes or the pathogens, but not on the human cells. These are present only on these pathogens or the microbes. These are found on the microbes, but on our human body cells, these are not found. It's PRR. Pattern recognition molecules are found on the microbial surface and not on the human cells. So the first one is TLR, that is toll-like receptor. So these are found on the surface and inside endosomes of the immune cells. These are found on the surface and the inside of the endosomes of the immune cells, like this. In the case of macrophages, dendritic cells. And their function is to detect this bacteria, fungi, and viruses by identifying their different signature, that is the lipopolysaccharides or the viral DNA or RNA. So this toll-like receptor will recognize the lipopolysaccharide layer of the gram-negative bacteria or viral DNA or RNA. They will look upon and call the immune cells for the action. So once these TLRs are active, they will induce the pro-inflammatory cytokines, and this type 1 interferons to fight from the infection. Like what these TLRs will do, they will initially recognize this bacteria, fungi, and viruses by their this lipopolysaccharide or their DNA or RNA presence in our body. Then they will call upon these immune cells and they will induce this pro-inflammatory cytokine production and this type 1 interferon production, and they will kill these pathogens. So the second one is RLR, that is RIG1 like receptors. So these are found in like inside the cytoplasm of the cells. Like these TLRs were present on the surface and inside the endosomes. And this RIG or this RLR are found inside cytoplasm of the cells. And their function is to recognize the RNA of this RNA viruses, and their response, like their work, is to induce this again pro-inflammatory cytokines and type 1 interferons, which will block the viral replication, which will block this RNA virus replication, and it will kill them. The next third one is NLR, that is node-like receptor. These are also found inside the cytoplasm of this immune cells. Inside the cytoplasm of immune cells, and they also detect these wide range of PAMP, that is pathogen associated molecular patterns, and their function is to induce again these pro-inflammatory cytokines and involve the processing of interleukin 1 family cytokines. So this node-like receptor will detect the intracellular bacterial components and other microbial this PMPs, and they will activate these pro-inflammatory cytokines and interleukin 1 family cytokines, which will kill this bacteria. So is this clear? There's three different type of pattern recognition receptors: TLR, RLR, and NLR. Is this clear?
Yes, no, it's clear. Yes, it's clear.
Okay, fine. So let's move to the week 5 assignment, the due date for which was 26th of Feb. So I hope you might have submitted this. So we'll check each and every question. The first question was to like what is their primary function of lysozyme in combating the pathogens? Like they are asking here the function of lysozyme that we have seen in our previous slides, that it was to hydrolyze the bacterial cell wall and leading to bacterial lysis. Like this is very easy and very direct question, so no need to think so much about this. The first main key function of this lysozyme is to hydrolyze the bacterial cell wall and do the bacterial lysis. So there is no point of lysozyme in promoting the vascular permeability. It have no function in that. It do not stimulate any kind of cytokine production. It also have no role of this antibody synthesis also, and it have no interaction with the T cell activation also. So its direct function is bacterial cell wall lysis. The second question is, what is the primary function of TLRs in the immune system? We have just seen that In the last slide, we have seen the primary function of the toll-like receptors is to rapidly detect a diverse array of pathogen associated molecular patterns, PAMs. This we will discuss in detail now. And it triggers the immune response, the innate immune response. So, what happens here if someone is not aware much about this TLR and their role? So, for that, I will quickly describe this again. Like these TLRs are a family of PRRs, that is the pattern recognition receptors, which are found on the immune cells like macrophages, dendritic cells, and neutrophils. And their primary role is to recognize the specific molecular patterns, which are the PAMPs, pathogen associated molecular patterns. And this each TLR is specific to detect different PAMPs, that is the different pathogen associated molecular pattern. Like there are so many different type of TLR. If you have seen the lecture slides in detail, you might be aware about TLR4, TLR2, TLR3, TLR5, TLR9. There are different type of TLRs which recognize different type of molecules on the pathogens. Like this TLR4 mainly recognize the LPS layer, that is the lipopolysaccharide layer on the gram-negative cell wall. And this TLR2 mainly recognize the lipoic acid on the gram-positive cell wall. Gram-negative, it will recognize gram-positive, and this LPS will be on gram-negative. TLR4 will recognize LPS on gram-negative. TLR2 will recognize the lipoic acid on gram-positive. This TLR3 will sorry. This TLR3 will bind to double-stranded RNA viruses. TLR5 is it will detect the flagellin protein found on the bacterial flagella. This TLR9 will it will recognize this CpG DNA. That is the unmethylated form. In genomics, you will study about the CpG DNA. That is unmethylated CpG DNA. So there are different type of TLRs and their function is different. Just a minute. We'll take 2 minutes break.
Okay.
So we were discussing about the role of different type of TLRs in recognizing different type of pathogen-associated molecular patterns. So we have seen that TLR4 is responsible for detecting the LPS in gram-negative bacteria. TLR2 is responsible for detecting the lipoic acid in gram-positive bacteria. TLR3 is responsible for binding to the double-stranded RNA in the viruses. TLR5 is responsible for binding to the flagellin protein in the bacterial flagella. TLR9 is responsible for recognizing the unmethylated CpG DNA, which is found in bacterial and viral genomes. So that these TLRs will scan for microbial signatures or these molecular patterns, and they will respond and call upon our immune system to quickly identify to the infection and respond accordingly. So the main function of this TLRs in immune system is to rapidly detect a diverse array of pathogen associated molecular patterns and trigger the innate immune response. So why these TLRs are very much important for our immunity? Because these can form a bridge between the innate and the adaptive immunity by activating the antigen presenting cells. Like these will identify these pathogens and they will activate the antigen presenting cells, that is the dendritic cells, and call upon the other immune cells. So they will also activate the adaptive immune system. So this form a gap or like this fill a gap between this innate and adaptive immune system. So that's why this TLR are important in the innate immune system. The next question is which known polymorphism of IRAK4 affect the TLR signaling? So for understanding this IRK4 effect on TLR signaling, we will see this. This I think you might have gone through in the lecture video also, but I will quickly summarize it here again. So what is this IRK4? This is a kinase in the toll-like receptor. This is a kinase that is responsible for the phosphorylation. It is a kinase in toll-like receptor and in the interleukin 1 receptor signaling pathway. Its main role is to activate the immune response against bacterial infection. So this IRK4, like here the question was this known polymorphism of IRK4 affect in TLR signaling. So answer to this was the polymorphism induces stop codons at 287 and 293 position, which increase the susceptibility to gram-positive bacteria. So like here we can see in the case of this IRK4, the stop codons 287 and 293. It increased the gram-positive infection. So what happened when the stop codon at 287 and 293 get mutated? Like these stop codons mutation means that the protein synthesis, it will end here prematurely, and it will lead to a non-functional or truncated IRA4 protein synthesis. So this will disrupt the further downstream signaling of this TLR and interleukin 1 receptor, which will make the immune system unable to respond to infections. Like this, if this IRK4 synthesis is truncated, so it is like premature here, like it is not in active form, so it is like non-functional. It is in its non-functional form, so it cannot start the downstream signaling. So it will disrupt the TLR and interleukin 1 signaling, because this IRK4 is mainly responsible for this TLR and interleukin 1 signaling. So on mutation in these two stop codons, that is 287 and 293. 287 and 293 stop codon mutation. This downstream signaling will stop here. So immune system cannot respond to the infections. So that's why this polymorphism introduced in the stop codons 287 and 293 will increase the susceptibility to gram positive bacteria, because the immune system cannot respond, this cannot function, so this gram positive bacterial infection will be increased in this case. So the next question is, what is the consequence of antibiotic treatment on commensal bacteria in the microbiome? Very easy and very direct, that it can lead to the dysbiosis. Here in the lecture slide, you might have seen this different type of interaction between these pathogens, like host and the microbe interaction. The first one is commensalis. The first one is this commensalism. Like here, microbe have a negative effect, like microbe have a neutral effect, and this host have a positive effect. Like host is happy, but microbes are on this neutral phase. So this is the commensal. So the next one is mutualism, which is the microbe is also getting some benefit and host is also getting some benefit. Both are happy here. And in parasitism, one partner is getting so much of this positive effects and other one is getting harm. It is the negative effect. So it is the parasitism. So here the cases of commensal bacteria in the microbiome. That is the consequence of antibiotic treatment on commensal bacteria. So what is here? So the answer to this is it can lead to dysbiosis. So the next question is, what are some characteristics of innate immunity that make it essential for survival? So you have to choose multiple options from this. The characteristics of innate immunity that make it essential for survival. So first of all, we have seen that this innate immunity acts as a first line of defense against microbes. It is the first responder to a pathogen attack, and it also initiates the immune response within hours of exposure. It is the immediate response from the pathogens. So, like in case of adaptive immunity, it takes so many days to create an immune response, but innate immunity is activated immediately on a pathogen attack. The next one is it compromises an ancient and evolved. It comprises an ancient and evolved system. Like this, innate immunity is highly conserved across species. So it is like we have this from our birth, so there is no differentiation to the pathogens or any kind of condition. So it is very ancient and very evolved system. Next one is it recruits effector cells to fight infection. That there are so many like this effector cells like we have discussed in cells of immune system. There are these neutrophils, macrophages, and dendritic cells which are responsible for causing the phagocytosis. So these effector cells are neutrophils, dendritic cells, and macrophages which cause the phagocytosis. Got phagocytosis of the pathogens. So this innate immunity will recruit these effector cells. And the next one is influences health through the microbial gene pool. So the microbiome and the innate immunity, they both interact to maintain our health. Like this gut microbiota and the innate immune system both interact well. So this beneficial or the good microbes will help in our immune system and it will protect us from the harmful pathogens. So these are the properties of the innate immune system. So the wrong options are it require prior exposure to pathogens for effectiveness. No, this is the case for adaptive immunity. Provides long-term immunity. No, it provides very short-term immunity because it don't have any memory. That is the case of this adaptive immune system. It adapts quickly to new pathogens. No, it is non-specific in nature, so it have nothing to do with the type of pathogen. So it responds in a uniform way to all type of pathogens. So these are the correct options for the innate immunity. The next question is MBL belongs to which family of immune proteins and what is its primary function? So MBL from the name we know this is mannose binding lectin. So from the name also we know that this is the sugar. So this is lectin family. It will bind to sugar on the pathogen surface. Like it will take this mannose as a EAMP on the pathogen. So it will recognize that sugars on the pathogen surface and it will bind to that. So this is the correct option to this. This is mannose binding protein. I'll write it here again. Mannose binding lectin will bind the PAMP on pathogen. So it is the primary function of MBL is to bind to sugar. And what about this other options? This is not an enzyme, so it will not degrade any type of microbial cell wall. It is not even a cytokine. It is not even a part of complement protein, and it is not also defensin family. So it is directly lectin family. So the next question is select all statements that correctly describe biochemical and chemical barriers involved in innate immunity. Again, about the innate immunity, that's why we have discussed today so many slides about the innate immunity because there were so many questions related to this. So the first one is this lysozyme break down peptidoglycan in bacterial cell wall because we have seen that this lysozyme is an antimicrobial enzyme found in tear, saliva, mucus. So this will break down the peptidoglycan layer in bacterial cell wall. The next one is mannose binding lectin binds to sugar on pathogen surface. Yes, in the last question we have seen that this mannose binding lectin will recognize the sugar on pathogen surface. Next one is alpha defensin and are antimicrobial peptides produced by the gut and lungs. Yes, in the case of this mucosal surfaces in the gut and the lungs, we have seen the role of alpha defensin and this antimicrobial peptides. These are the this cationic peptides, that is the this positive plus charged peptide which interact with the this antimicrobial peptides or the pathogens of this negatively charged in the epithelial cells of this lung. So they will disrupt the membrane integrity and it will kill the pathogens. Next one is cathelicidin is an antimicrobial peptide stored in lamellar bodies. Yes, we have seen that these cathelicidins are the host defense peptides which are found in this many type of immune cells like neutrophil, macrophages, and these are stored in this lamellar bodies in the skin and the mucosal tissues, and these are released upon the infection to an pathogen. Next is histatins are antimicrobial chemicals present in the oral cavity. Yes, these histatins are again the cationic this molecules, that is the positive charge containing molecules, and they will also interact with this negatively charged pathogens. So these are the correct options, and if we discuss about the wrong options, then we are discussing here about the innate immunity. So the wrong option is complement protein act only as a part of the adaptive immune system. So these complement proteins also function primarily in the innate immunity, but it also support the adaptive immunity. So it act only as a part of, no, it comes in the case of both these things, both innate and adaptive. The next is NK cells directly form part of the chemical barriers against pathogens. No, these are NK cells are not chemical barriers, these are cellular barriers. The next one is CRP, that is C-reactive protein, is a pattern recognition receptor on immune cell surface. On immune cell surface. No, this is not on the immune cell surface. So this is also wrong. Now next question is the term pathogen associated molecular patterns, that is the PAMP that we are discussing from so many slides. These refer to. So these are the molecules that are shared by different pathogens. Like this PAMP. What are these PAMP? These are the conserved molecular structures found on different type of pathogens. Like pathogens will be different, but their patterns will be same. So they will be shared by different pathogens. And they will not present on the human cells. So these are unique molecules found only on the host cells, no on host. They will not be present. They will be only found on the pathogens. So these are the molecules that are produced by the host in response to the pathogen infection. No, these are the patterns present on pathogens recognized by host cells. The molecules that are produced by the adaptive immune system, no, these are not produced. Molecules that are only found in viruses, no, they can be found in all type of pathogens. So there is a similar terms, that is PEM and the PRR. So do not get confused between these terms. Like this, PEM are the pathogen associated molecular patterns present on the pathogens. And these PRR are the pattern recognition receptors which are found on the host cells. So what is written here, upon PAMP recognition, PRR activation transmit intracellular signal, activating a multitude of intracellular signaling pathways, ultimately result in the activation of gene expression and synthesis of a broad range of molecules, including cytokines, chemokines, cell adhesion molecules, and immunoreceptors, which together clear out the pathogen from the system or can activate adaptive immune response. So upon PAM recognition, this PRR will activate the intracellular signaling. Like this PAM will be present on the pathogens. Like this lipopeptide, it is a pathogen, it is present here. This PAM and this PRR are the pattern recognition receptors present on this host cell. This is here. So this PAM will interact with the PRR. PAM will interact with the PRR. PAM is present on pathogen and PRR are present on host cells. These both will interact and on their interaction, the downstream immune signaling will start. An immune system will produce so many like these chemical molecules like the cytokine, chemokine, and many other. And it will kill the pathogen. So do not get confused between these both terms. This PEM is present on the pathogens, not on the host cells, and this PRR are the receptors present on the host cells. So don't get confused between these both. So this PRR and PEM, difference between both these types. PRR present on the innate immune cells at cell surface receptor or cytosolic PRR, and these PEMs are evolutionary conserved structures on the pathogens. These are present on innate immune cells. This PRR can distinguish cells and non-self cells by recognizing PEM. So these PRR will recognize this PEM. And these PEMs are the signatures present only, mark it here, present only on the pathogens. And these PRR are generally glycoproteins, and this PEM can be carbohydrate, protein, nucleic acid, or anything. These can be like literally anything. And this PRR are the glycoprotein because these are the receptors on the host cell. And this PRR include like this TLR, and this TLR are membrane bound receptors localized at the cellular or endosomal membranes. This PEM include the flagellin protein present in the bacterial flagella, lipopolysaccharide layer of the gram-negative bacteria, peptidoglycan layer of the bacterial cell wall, zymogen of yeast cell wall, nucleic acid of both bacteria and viruses. So these are the different type that can be found in different type of pathogens. Like they can be either flagellin protein, they can be LPS layer, they can be peptidoglycan layer, they can be zymogen, they can be nucleic acid, they can be anything. And the next one is this cytosolic PRRs is include retinoic acid inducible gene, that is RIG1, like receptor, that is RLR. Like this PRR include this TLR, RLR, and this NLR. This include this three type of receptors. And here the nucleic acid of virus and this bacteria generally interact with cytosolic PRR. So interact mainly with the cytosolic PRRs. So don't get confused between this PRR and this PEM. PRR are present on host. These PEM are exclusively found on the pathogen surface. So don't get confused. So some examples of PRR and PEM interaction. So in the case of this gram-negative bacteria, which is recognized by TLR4 via the lipid A portion of the LPS. So here this TLR4 is the PRR and this lipid A portion of the LPS is the PEM. And the next one is lipothioic acid, lipoprotein, and peptidoglycan of gram-positive bacteria are detected by TLR2. So again, here this TLR2 is the PRR and this lipothioic acid, lipoprotein, peptidoglycan are the PAM. Again, the flagellin, the major constituent of the motility apparatus of flagellated bacteria, is recognized by this TLR5. So this TLR5 is here PRR and the flagellin is the PAMP. And this TLR3 recognizes intracellular double-stranded RNA produced during viral replication. So this PRR recognize this PAMP. And this TLR7 and TLR8 are activated by single-stranded RNA viruses. So these are the different examples where this PRR and PEMP interacts. So I hope that is clear. There is a lot of in-depth discussion about this PRR and PEMP because this is major part of this particular concept. So don't get confused in this. The next question is, which family of pattern recognition receptor is primarily responsible for sensing RNA viruses? That is the RRG-like receptor. So here we have seen that this RRG-like receptor, that is the RLR, it recognizes the intracellular sensor recognize RNA of RNA viruses, and it induces the pro-inflammatory cytokine and type 1 interferon. So it will exclusively recognize the this what they are asking here, responsible for sensing RNA viruses. So it will recognize this RNA viruses. The last question was, what is the significance of the TIR domain in toll-like receptor? So like it is essential for downstream signaling after the ligand binding. So what was this TIR receptor? That is the TIR domain in toll-like receptor. So it is the, I'll write it here, toll interleukin 1 receptor. That is TIR. This is a domain of TLR. That is TLR is the receptor and the toll or interleukin 2 receptor is the domain of this TLR, which is required for the immune signaling. So these TLRs are again this TLR are PRR pattern recognition receptors, which will recognize the PEMP of these pathogens. So this TLR domain will cause the downstream signaling. So this is the main domain where the this PAMP will come and bind. So it is the main domain. So it plays a critical role in transmembrane insertion. Node is responsible for binding to the ligand. It is responsible for recognizing PEMP. It will not recognize. It is required for the binding and the downstream signaling. It mediates the cleavage of TLR for activation. No, there is no need for cleavage. It is involved in the recognition of lipid components. No, the correct answer is it is essential for downstream signaling after the ligand is bound to that. So these were the questions from your week 5 assignment, and till now these topics are covered on the NPTEL portal, so you can check it out there. And on next Saturday, we will discuss about the week 6 assignment. So, till here, if you have any doubt or any query, you can ask me now, or else you can post it on the discussion forum of NPTEL, or you can also send me an email if you have any query or any personal topic you want to discuss. So, you can drop me an email and do check out the YouTube channel where this video will be uploaded. If you have any comment, then you can let me know. Is everything
Thank you
clear
so
till
much,
here?
ma'am. As it is crystal clear, and today's lecture was really amazing, ma'am. Thank you so much.
Okay, thank you so much. It means a lot. So, if you have any doubt, you can write in the chat box. I will take your queries from there.
Yes, ma'am. I'll just go through it once again later, and if there are any queries, I'll just can I just mail you and we can discuss about it. If there are any,
Yes, sure. You can drop me an email regarding your query. You can go to the slides again and also the YouTube video. You can visit again and let me know.
okay. Thank you so much.
Okay. Thank you.
Can I leave now?
Yes, sure. Thank you for attending and you can leave.
Yes, thank you so much.
Okay,
Thank you so
thank
much.
you. Bye bye. Happy learning. Okay.